as I do. All righty. All right, let's get started then. At any point today, and from here on out, please stop me if you're like, that didn't make sense. Please repeat that. Or can you go back and explain that a different way? Because I want you guys to understand this stuff rather than just be like, well, she said it once, I just have to suffer with that explanation. Okay, so let's get started with the structure and the function. Hopefully a lot of this is review. I have a couple of the videos posted from YouTube for you to watch as well if you're like, I don't really remember passive transport and all that stuff. But I'm going to try to go over as much of that as we have time for today as well. All right, so I am going to be asking for your input as well. Oh no, why is this not working now? Technology, man, you can never rely on it. There we go. Okay, just got to click on the stupid screen. All right, so these are the objectives. If you ever want me to go through them, let me know, but they're more there for you when you're reviewing. We got a ton of key terms this time, but it's all good stuff. So starting off, most cells in general and most bacteria share these fundamental traits. So for bacteria, they have very thick envelopes and they're very unique and they're there to protect the cell. They have very, very compact genomes. The majority of a bacterial cell is DNA. And that is very different from eukaryotes. Yeah, I am, yes. And there's also very tightly coordinated functions. We're going to be going over this more, especially when we get into the genetics, but these cells are very tightly oiled machines. Things happen in concert with each other that don't normally happen all together in a eukaryotic cell, and they have this down to a literal science. Now, archaea are like bacteria in that they're prokaryotes, but they're going to have a lot of different unique changes. So they're going to have adaptations that help them survive their extreme environments that make them a little bit different from the bacteria that we're going to talk about. And just as a reminder, we're not really going to talk about eukaryotes, even though there are microbes that are eukaryotes, but they've got a nucleus and they have membrane-bound organelles, and prokaryotes do not have those things. Now, this here is a really great picture because a lot of times we look at a cell and we're like, oh, it's just a lot of water and empty space. And then there's like DNA in a little spot of it. No, these things are packed full of chemicals at any moment. They're very busy. They're very complex. They're highly, highly organized structures. So this is a more realistic image of what is the inside of a cell is going to look like. Any questions? Right. So what do all cells, whether they are a prokaryote or a eukaryote, bacteria, archaea, or eukaryotic, what do they all have in common? Well, they have a cytoplasm. You might have also heard the name cytosol. Those are technically two different things. The cytosol is the liquid portion of the inside of the cell. The cytoplasm includes all the dissolved stuff inside that liquid as well. But for our sakes, we're just going to use the term cytoplasm to indicate both. It's the gel-like fluid that has all the dissolved solids and all the dissolved nutrients that are in that liquid. Then every single cell has to have genetic material to dictate and organize what happens in the cell and what type of cell it is. You need to have ribosomes because we need a way to turn that blueprint of the genetic material into a physical structure. That's what ribosomes do. They take the DNA and they turn it into proteins that build the structure and do functions for us in the form of enzymes. And then they have a cell membrane because you're not you unless you are separated from everything else. You need to actually say, this part is me, that part over there is you. So we need to have a separation or a barrier between you and the outside world. That's all review still, right? Okay, good. Now let's go more into bacterial cells. They do have that cytoplasm and the cell membrane, but they'll have a cell wall. All bacteria have cell walls that are highly, highly complex, and that's what we're getting into for the majority of today. That's going to be a big portion of what's on the exam. They have a nucleoid. Since they don't have a nucleus, all their DNA is kind of condensed into a central area of the cell. It is actually connected to the membrane itself. So at the origin of replication, it sticks to the membrane. And then they form a really cool coiled loop structure because their chromosome is just one circular chromosome. And they will loop it to organize it, and it looks like a little daisy inside the cell. It's actually quite pretty. And then some of them will have a flagellum, which we'll talk about at the end. And that's just this long tail-like structure that helps it swim or move around. Not every single cell that's a bacteria has a flagellum, just some do. These are the typical ones that you might find in a bacterial cell. So any questions about that? Okay. Seriously, stop me anytime. Not all of them do, no. Well, that's going to be an accessory structure, kind of like the flagellum. They all have these ones: cytoplasm, cell membrane, cell wall, nucleoid. The flagellum is just a very common extra piece that they have. The pilus, the capsule, all of those are extra things that some will have and others don't. Good question. All right, so cells also all share similar chemical components. 70% of basically every cell on the planet is water. Our cells, bacterial cells, we are majority water. Why? Let's go back to chemistry here. Why do we need so much water in our cells? Not quite, but I like the thinking. Well, we have a lot of polar molecules that we need to be able to move around. Yeah, so that's very close to where I was going with this. So we have a lot of water because the majority of reactions required for life have to be dissolved in water. They're aqueous reactions. So we need water to facilitate all the chemical reactions that keep us alive. If we don't have that, these chemicals won't react with each other and we will not exist. Then we have essential ions. These are things like sodium, magnesium, chlorine, all that fun stuff. There's small organic molecules. The ones that are relevant to what we're going to talk about with bacteria are going to be a lot of your weak acids and weak bases like lactic acid, acetic acid. We'll talk about those a lot. And then we have the macromolecules. We've also heard these as biomolecules. What are the 4 biomolecules? I know, I'm making you dig deep back to first and second semester of bio. What are the 4? What? Nucleic acids, I heard that. Yeah, that's like our DNA, RNA. Nucleic acids is one. Lipids are fats, yep. Carbohydrates, the sugars. Last one. Proteins, yes, that make up our structure and our enzyme, right? So that's the other major portion of every cell, are those 4 different molecules that make up the cell, dictate the actions of the cell, and all of that. Now, the composition of these things, how much of one or the other of these 4, it really depends on the cell. It depends on the species. It depends on what growth phase the bacteria are in. It depends on the environment. So there's a lot of asterisks when it comes to exactly what percentage of the cell is made up by each of these 4, but they have to all be there in the cell. Yeah. It definitely can. Yeah, the pH will affect kind of the stability of the membrane or of the cell wall and membrane. It'll affect what types of ion gradients we can make and how effective they are to store energy for the cell. So yes, any type of environmental condition can have a drastic impact on how the cell functions. And we'll get into all that too. Not quite at a deep level. That's, if you're into that kind of stuff, I recommend you take microbial physiology after this. That's a great class to really get into the cool exceptions and how the environment affects the bacteria. All right, we're going to first talk about the plasma membrane. And I'm going to start it off with still putting you guys on the spot. What do we remember about the plasma membrane? Lipid bilayer. What does that mean? You don't have to answer unless you want to. What does it mean that it's a lipid bilayer? Selectively fully permeable means anything can get through. This is selectively permeable. Only the things that it wants to pass through can pass through. What does it mean that it's lipid? It's majority of really 50% fat. And that's going to affect what type of things can move through and what is the primary two structures that make up the membrane. Do you guys remember? That's the hydrophobic philic head and the hydrophobic tails of the phospholipid. Yep. And what are these other structures made of? Do you remember? Close. They make these. Ribosomes make these things. Proteins, yeah, we have membrane proteins. So it's lipids, mainly phospholipids, and proteins. So what we mean by bilayer as well is these phospholipids will naturally form two layers in the cell. And let me get my marker because I'm going to draw this out for you. Okay. I really want you guys to understand the basics because if you're not strong on those, we can't build off of it. So that's why I'm hitting this hard. So the phospholipid has a hydrophilic head and two hydrophobic tails. This is polar, and these are non-polar. Do we remember what those terms mean? Yeah, I'm going to review it just in case you're like, I did not answer yes, but I'm not going to say no out loud. Polar means it has a charge. Either there's an area that's partially positive and partially negative, or we're talking about an ion where it's literally fully positive or fully negative. We lump those in polar. That's also what we call hydrophilic. These are things that, like water, they can be dissolved by water, they can be involved in aqueous reactions. Non-polar, these are things like our lipids, our fats, our oils. They have no charge differential in them. So all of their hydrocarbons are going to be neutrally charged. Because of that, water can't attack it anywhere. Water can't dissolve any portion of it. Therefore, they are hydrophobic, water-fearing. They don't have any interaction with water at all, and they try to get away from it. So if a phospholipid is randomly floating around in the solution of the cell, it is not going to want to stay like this. It's going to want to get this hydrophobic portion covered up and away from the water as quickly as possible. So as a result, they naturally form, oh no, they naturally form this bilayer where the hydrophobic tails face each other and are protected from the aqueous external environment and the aqueous cytoplasm by the polar heads that like to interact with water. So this is a natural formation that the cell will take automatically. If it's dropped, the cell membrane will take automatically in an aqueous environment. Now also, we call each of these layers leaflets. And that's going to matter more in microbiology than in eukaryotic cell biology that you learn, because we're going to have more than one membrane in a moment. So this will be the inner leaflet. This will be the outer leaflet. And technically, there's going to be a difference between those. We want our inner membrane to have slightly different characteristics than the outer membrane leaflet because they're going to be interacting with different structures. So we want the ones on the inside to maybe interact with our DNA, the ones on the outside to be able to interact with other cells. So the inner leaflet and the outer leaflet will be slightly different from each other. Yes. Yeah, so this is one leaflet with the heads facing down, the tails facing up. The second leaflet, the outer leaflet, is this one right here with the heads facing up and the tails facing down. Good question. All right, so I'm going to repeat myself a little bit now. I like going over the pictures first because this is a great way to learn how to study biology. Look at a picture and describe every aspect of it. If you can't do that, you're not ready to take the test yet. So now let's put this to words. So the purpose of the cell membrane is to separate the cell from the external environment, to protect the internal structures. It strictly manages movement in and out of the cell. That was that selectively permeable term that we heard. And then it also can store energy. We're going to talk about this more next class. But what it can do is move a bunch of ions to one side of the membrane and kind of use that storage as like a battery. And then it can open the floodgates, let that charge difference flow through and make electricity and use that to power the cell. Then it's also an anchor point for the chromosome. This is something that prokaryotes do that eukaryotes don't. It's equally comprised of phospholipids and proteins, and by that I mean weight. So if you weigh out the membrane, 50% of it will be phospholipids by weight, 50% proteins. There are technically more phospholipids, they're more numerous, but proteins way more. All right, I'm gonna move on, because a lot of that I just said twice now. And you do have all the slides, remember that. So the phospholipid, this guy here with the crazy little head that looks like a clothespin, this is the most common type of lipid in the membrane itself. It has the two hydrocarbon tails. It has an ester link, which is going to come up again later. You don't have to understand the chemistry of that. Just know that because it has this double bond to the oxygen, it's more likely to be dissolved by water than other things. Then it attaches to a glycerol, which is attached to a phosphate group. And that phosphate group is very, very negative. So these phospholipid heads have a highly, highly negative charge. So in general, the top and the bottom of all your membranes will be negatively charged. And so they'll naturally form that bilayer based on that difference between the hydrophobic tails and the hydrophilic heads. And then they each form the two leaflets naturally in the membrane itself. You will not have to draw this. So I'm not one where it's like you have to understand the chemistry. That's for chemistry class or biochemistry class. You have to understand the pertinent chemistry. You don't have to draw me anything. And you'll understand how this is important later. You have to understand that these are hydrophobic. That's technically a chemistry thing, and that this is charged, therefore it's polar. But that's the extent of the chemistry you really have to understand. All right. Now, because of that charge group, sometimes the cell doesn't want the phospholipid head to be negatively charged. So what it can do is on top of that phosphoryl head group, it can add other functional groups. And in this case, it added an amino group, which is positively charged. The cool thing about this, when they change it to a positively charged head, this is going to allow the phospholipid to have new functions that other ones don't. In this particular case, it allows it to interact with DNA. So if you're looking at a membrane and you see an area where a lot of the phospholipids are highly positively charged, you know it's going to be interacting with something negative there. And DNA is highly, highly negatively charged. So whenever we add positive charges to our phospholipids, expect that to be an anchor point for our DNA. And then because of that, we end up with the different compositions or makeup structure of the different leaflets. Sorry, sometimes I get excited and my words in my head go faster than I can speak them. I love this stuff so much. So I apologize if I get a little nerded out sometimes. Okay, any questions about this before we move on? Is this finally helping you understand a little bit more? Okay. I'm the person that's always like, yeah, but why? But why? But why? I can't understand the whole until I understand the steps that lead up to it. So if I give you a lot of background information, that's why. I'm trying to give you the whole picture. All right, so what are the functions of these membrane proteins? Because we got a lot of them. Well, first, they can offer structural support. They can attach to cytoskeletal filaments inside the cell and kind of anchor parts of the membrane in place. They can detect environmental stimuli or signals, and that way the cell can say, hey, there's food out there, or there's a dangerous chemical building up in the environment. We got to get out of Dodge. They can communicate with other cells. They can secrete chemicals that are they can be passageways for chemicals to be released into the environment. Some of these we call virulence factors. They're things that then end up attacking our body during infection and make them more dangerous to us. And a big one that they do is ion transport and storage. And a lot of these are not just one protein. What will happen is many proteins will be made and they will be brought together to form a larger machine. And we call that a complex, a protein complex. So here, this is ATP synthase that makes ATP. It's not just one protein. It is many, many protein subunits coming together to form this machinery. This is the protein complex that I worked on. And it has an ACRB component, which has 3 ACRB proteins connected to 3 ACRA proteins, connected to 3 TOL-C proteins, and then there's 3 ACRZ proteins in there too. And they all work together to capture antibiotics and spit them back out of the cell. This is kind of like a hole in the membrane that allows things to pass out of the cell easily. We have tons of those holes in the membrane that we call porins. And they're literally, this part will sit inside the membrane itself, like right here, and it'll just be a hole. And that way things can pass in and out of the membrane rather than passing directly across it, which we'll talk about why that's needed in a moment. So lots of functions of these proteins. Now, because they're going to be embedded inside the membrane, our membrane proteins have very, very specific structures. And this picture is actually really cool. So red indicates positive charges on the protein. Negative charges. Brain flipped it. Blue indicates positive charges in the protein itself, and white indicates neutral or no charge. So in this part of the membrane where we have all of our hydrophobic non-polar tails, it makes sense that we see a lot of white, a lot of neutral charges in our protein. Because if they're charged parts of our protein, that means they're polar. That means they want to be around water. They're hydrophilic. So they won't like being in this membrane portion. Naturally, they'll want to go outside into the environment. So if you want it to stay inside the membrane where the tails are, you got to make it nonpolar. Then we have a lot of positive and negative charges on the outside and kind of buried within the heads of the phospholipids. That allows the protein to literally anchor itself to the phospholipid heads. If the phospholipid heads are negative, we see a lot of positive charges in our protein, so they interact, and literally those positive and negative charges bind together to anchor that protein in place so it doesn't move around. So here we have the charges because we're dealing with polar environments and aqueous environments, and here we have the non-polar. And it's really cool when you look at the sequence of these amino acids. You'll see a portion that has a lot of charged amino acids, then a portion that has no charge in the amino acids, then a portion with charges, then a non-polar charge. And what will happen is the protein will feed itself through the membrane like this, in kind of an up and down threading motion. And it really embeds that protein in there well. And this is just a really cool example of a co-transporter. This particular protein helps transport leucine into the cell, which is an amino acid. So, any questions about that? I know it seems like, what does this have to do with bacteria? But this stuff is going to follow you through every biology class. So the more you practice these basics and really understand them, the more you'll get to complex things and say, oh, that's why it's behaving this way, or that's why it has to have that structure. Or you can look at the structure of something and say automatically, that must be found in the membrane, just based on how the charges are divided. So it's why we're going through all these basics. Now, these are called amphipathic molecules. Amphi means both, pathos means experience. So this is a molecule that has the experience of being both polar and nonpolar, just like our phospholipids. They have, I just realized you guys over there can't see these phospholipids. I'll draw them here too. These have a polar and a non-polar portion. They are experiencing both experiences at the same time. They are amphipathic. You will hear that term brought up a lot more in your higher level classes. Okay, so moving on, we're going to review how things get across the membrane and kind of a little bit of a crash course here. So I'm going to go a little fast, and if you have questions, there's videos posted, or you can always ask me and I can explain it after class or during office hours. So things move across the membrane through what we call passive diffusion. This is where anything very, very, very small, anything that is non-polar or uncharged, they can actually directly pass through the membrane. They kind of like squeeze in between the phospholipids and like swim their way through, kind of. This is stuff like oxygen and CO2. Very, very small, overall no charge to these molecules. But larger or charged or polar molecules, they need proteins to cross. They can't just muscle their way across here because they're technically always surrounded by water. They're in constant dissolved states with the water. So in order for them to cross, they'd have to bring the water with them. And water doesn't like to be in that center point of the membrane. So they will just pass through a protein that creates a channel for them where they can bring the water with them. This is called facilitated diffusion, diffusion with the help of a protein. Now these things happen, this movement happens completely naturally. There's a physics phenomenon called entropy, which basically says the universe wants things to be chaos. The universe does not like order. Having lots of things over here and none of them over here upsets entropy. It says, I just want chaos everywhere. So because of that, if we have a high amount or a high concentration on one side, naturally those molecules are going to move across to the other side where there's a lower amount, a lower concentration, until it's equal on both sides. Then that's a perfect state of maximal chaos because everything's equal everywhere. So we call that a concentration gradient, when there's more on one side of the membrane than the other. And with diffusion, it moves from a naturally high to a naturally low state until equilibrium is achieved. So when this happens, we call it passive transport because that requires no energy. This is literally happening because every molecule is constantly vibrating and in motion, and we call that Brownian motion, and they will just naturally kind of move around until they get into this perfect equilibrium state. Now, if we want to move from a low concentration to a high, if I wanted to take these molecules and push them back to the other side, that's going to require energy. So we call that active transport. So think about it this way. If I were to spray a can of Febreze here, there would suddenly be a really high concentration of the scent particles on this side of the room. Give it time, and they will naturally passively move without us doing anything to the whole room. But then if I told you guys, okay, bring all the scent particles back and put them back in the can, that would require a lot of energy on our part. So no energy for them to passively move, lots of energy to bring them back. So just a kind of a visual representation of that. Any questions about passive or active transport? All right, let's move on then. So one form of passive transport is called osmosis. And we don't just call this passive diffusion because it's water. Water's super special. There's so many cool things about water. You guys really have to look into it and have fun learning about water. But water is technically polar, but it is also very, very, very small. So technically, water can naturally diffuse across the membrane. It just does so very, very slowly. So in this case, the movement of water across a membrane through passive transport is called osmosis. And the problem is that whenever that happens, it usually will bring dissolved substances with it. So that's why it's a special case. Everything else diffuses based on its own gradient, its own concentration gradient. Water diffuses based on how much stuff there is surrounding it. It wants to go where there's less water, but that's also going to affect the concentration of all the dissolved stuff inside. You don't have to really understand that for this case. What you have to remember are the environmental conditions. There was hypotonic, isotonic, and hypertonic. Do we remember those? At least the words. So in a hypotonic environment, this is what most bacterial cells exist in. They have more water external to the cell in the environment. So if the inside of the cell is 70% water, maybe they're living in an environment that's 90% water. Based on the laws of entropy and diffusion, that 90% is going to move into the cell where it's 70% until it's equal on both sides. So that's going to create a lot of pressure inside the cell. Think about it as filling up a water balloon. The more water you pack into that balloon, the more pressure you're putting on the balloon itself, and it's going to stretch and stretch. If you put too much water in there, it can explode. That internal pressure pushing out is called osmotic or turgor pressure. And if we don't control it, the cell can burst. It's actually a mechanism that a lot of our antibiotics use to kill the cell. So what bacterial cells use to keep themselves safe is they put a cell wall on the outside. That cell wall, the whole job of it is to keep the cell from exploding because of turgor pressure, basically. So hypotonic is a state where water is rushing into the cell. Isotonic is where it's equal in and out. And hypertonic is where there's more water inside than out, like dropping one of our cells in the Dead Sea where it's 30% salt. We will start losing water like crazy, and so our cells will shrivel up. A good way to remember it, just because I struggled with this, I couldn't keep the term straight. This is going to be really silly, but I promise you'll never forget it again. Hypo-OMG, I'm growing. Hyper, I'm shrinking. Yeah, really crazy. But like I said, you're going to remember it now. And then I am isotonic. The idea that we create an isotonic environment for our cells, so we're constantly in a good equilibrium. Okay, well, yeah, go ahead. No.
So passive diffusion and passive transport are different.
No. Passive diffusion is a type of passive transport. Facilitated diffusion is also a type of passive transport. So they're kind of interchangeable. Passive transport just means whatever transport is happening requires no energy. And diffusion just means it's moving across without energy usage. Yeah. Good question. Yeah. So we'll talk about this more next class, but the membrane allows for the storage of energy through creation of these gradients. So what the cell will do, particularly with hydrogen ions, we also call these protons because the hydrogen atom is one proton, one electron. If it loses an electron, it's just a proton. So what it will do is take these hydrogen ions and pump them all to one side of the membrane. So we create a gradient. Now there's a ton of it outside of the cell and none inside. And it's basically a battery stored up for later. Then we can use different proteins to allow them to passively move back in. And that movement of charged particles is basically electricity. So it's powering the cell to also move something against its gradient. We kind of capture the energy of this moving through the protein to also move something from low to high concentration too. So we use that energy and couple it with other transport to save the cell energy in the long run. We will definitely talk about this more later. We call these types of transport proteins transport proteins or transporters. Next class, we'll talk about like co-transport, sim transport, anti-transport, all those other things. But right now, you just have to know we make these ion gradients to store energy. Any questions about that? This is all somewhat familiar to you guys, right? Maybe you haven't heard it in quite this way, but you're like, oh yeah, I know that. All right. Here's an interesting case. There are these things called membrane permeant weak acids and bases. So an acid is something that gives up a proton, gives up a hydrogen ion. And a base is something that takes up a hydrogen ion. And these are very highly charged. They will be hard positive, hard negative charges when they dissociate. And they're almost always dissociated. Weak acids and weak bases exist in an equilibrium form. They will have their stable, uncharged form, and they will have their dissociated form. And when they're in an aqueous or water-based environment, they will naturally go back and forth between the two structures. So these ones, unfortunately, can enter the membrane when the membrane doesn't want them in there, because they can switch into their neutral form, glide across the membrane because they're uncharged, and then when they get back in, they can dissociate and increase the acid inside the cell, even if the cell doesn't want it, or increase the base in the cell, even if the cell doesn't want it. So unfortunately, this can result in acid or alkaline stress on the cell, and the cell will have to take steps to mitigate this. Sometimes it can't, and it'll die. So examples of these are like acetic acid, lactic acid. Those are going to be important when we talk about metabolism later. But other cool ones, aspirin, penicillin, or Prozac, or tetracycline, the antibiotic. These can all just pass through the membrane easily, which is kind of a good thing. We don't want to have to make our aspirin fight to get inside our cells when we want them. And this is for all membranes, not just bacteria, because all our membranes are basically the same. Now, here's a question for you. Do you know how fermentation works? We know bacteria do it, but do you know why we do fermentation and how that whole process works? Anyone have an idea? Why do we do fermentation first? To what? Preserve food, what did you say? Yeah, it's in the absence of oxygen, and we use it to preserve our food because back in the day, we had no refrigeration. So how did we keep our fruits and veggies from going bad over the winter? We needed to preserve them somehow. We ended up either salting them or preserving them with bacteria. And what this does, you take the bacteria in the food and you put them in a closed container. So the waste products are going to build up and build up and build up. The food is going to get less and less and less and less for these bacteria over time. So what happens is, as the bacteria are naturally eating the food that you're eventually going to eat, they start breaking it down through metabolism. This is going to be anaerobic mostly, so they're going to start producing a lot of acetic acid, lactic acid through fermentation methods. These will then just start to come right back inside the cell, and the cell will try to fix that problem, but these waste products, these membrane permeant weak acids, are just building up in the environment, and it's going to get to a point where the cell can no longer fight off that acid stress, and all the bacteria die and they stop being able to eat your food. So that's why when you have fermented veggies, they're not quite as crisp as they were when they were fresh. They're kind of like they got cooked a little bit because they were technically being cooked or eaten by the bacteria, but then all of these weak acids killed anything that could possibly be living inside there and prevented the food from further spoiling. Because food spoilage is just because bacteria is eating it, and they end up making it go bad because they're eating it all. That's okay. Yes, it's the exact same thing. Yep. 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 So sourdough is a type of fermentation. I cannot remember the acids that are produced in that one. I'm sorry, I'd have to look into it again. But instead of bacteria doing it, it's a single-celled eukaryote that we call yeast. Yep. Same thing with really any bread. We cook it with, we have it, you know, the yeast eats the bread itself, the flour, and makes it more tolerable for our stomachs. And it also helps create all those beautiful air bubbles in the bread. Otherwise, the bread would be super dense because they also produce CO2, which is also highly acidic when it's dissolved in water. So we end up creating this really acidic environment whenever we do pretty much any kind of fermentation. Yeah. Good questions. Yeah. These acids will rush into the cell and the acid stress will kill the cell. So they eventually die because of their own waste products killing them. So let's go back to the membrane structure, because we talked about the typical membrane structure. Let's talk about some atypical structures now. It's ones that the cell will do in order to survive extreme stresses or starvation or things like that. The first thing is that they can make alternative phospholipids. One of them is called cardiolipin or cardiolipin. I've heard it both ways. It's a double phospholipid where the heads are combined with a glycerol. And it makes this V shape here. And what this does is it increases fluidity of the membrane so that your phospholipids don't pack together as closely. Let me give you a visual example of what I mean by that. In an elevator, if everyone stands perfectly straight, you can pack a lot of people into that elevator. But as soon as everyone kind of starts leaning like this against the walls and they have like a curve in their body, they're taking up the spot of where 2 or 3 people could potentially stand. So you can pack fewer people in there. When you're packed in super tight, you can't really move around a lot. You're going to have like a very stiff posture in that tightly packed elevator. But when you're all stretched out, you can kind of maybe move around a little bit. You'll have enough space to maybe switch places with someone else in that elevator. Same thing with our membrane. The membrane is constantly going to be trying to find a balance between tightly packed and looser and fluid, because we want to allow some things to get through easily, but sometimes we don't want things to come in if we're in a dangerous environment. So we want to make the fluid more stiff to prevent things from being able to diffuse easily. If there's plenty of food and it's a really great, safe environment, we'll make the membrane a little more fluid so things can pass through easily. So with this, the V shape helps increase fluidity, but the more important thing with the cardiolipin is it helps determine the cell shape. We've labeled it with fluorescent dye, and we notice that it likes to congregate on the ends of cells, where it causes the membrane to take a really sharp turn. So in cases where the cell is starving, it will start to make more and more and more cardiolipin, and it'll make the cell get smaller and more rounded. A smaller cell is easier to keep alive than a really big one because it needs less nutrition. So by adding these, it reduces the shape of the cell to a smaller, more manageable level to survive the starvation conditions. So that's the idea behind switching up and adding this particular phospholipid. It also was found to bind some proteins that help protect the cell from osmotic stress. So if it's like, we're taking in a lot of water, that's a lot of osmotic pressure, or the opposite, it'll help protect them from that influx or loss of water. Any questions about that? Okay. There's not just cardiolipin though. We have lots of other things we can do, and this you've probably heard before. So hopefully mostly this is review. Do you remember that fatty acids can be saturated, unsaturated, polyunsaturated? What did those mean? What's a saturated fat? All single bonds. So like this one here, the points are carbon, all of them between the carbons are single bonds. And we also have a bunch of hydrogens coming off them with single bonds too. We just don't show those for simplicity's sake. Are saturated fats more solid or more liquid at room temperature? Solid, yeah, those are our animal fats and things like butter. So it's more solid at room temperature. But what we can do is we can add a double bond in between some of our carbons, and that makes them unsaturated. Saturated means they have as many bonds as possible to hydrogen around them. By causing a double bond between 2 carbons, we now have less room for hydrogen to bond to those carbons because they have 2 bonds now occupied already. So it's not as saturated with hydrogen as before. That's where the term comes from. When this forms naturally, what happens is the hydrogens that are bonded will bond on the same side. We call that the cis configuration. Cis means on the same side. Well, that causes a bend in the molecule because those hydrogens are pushing away from each other. So it causes the whole fatty acid to bend, and that creates more fluidity in the membrane. So unsaturated fats are more liquid at room temperature than solid. Now, we found a way to turn these into trans fats, which make them back into the straight form. And these, our body doesn't really know what to do with them. So they tend to make them just kind of pack on your heart and start to solidify in your heart, and you get a heart attack. So that's why we say try not to eat too many trans fats. And they're in a lot of those highly processed foods, unfortunately. So these saturated fats will cause the membrane to pack very tightly and be very stiff. The unsaturated fats allow for more fluidity. Then sometimes we can even create really special ones. We can create these cool 3-carbon cycles, not cycles, that's not the word I'm looking for. I'm sorry, my brain, rings, oh my gosh. And what this does, it kind of acts in the same way as an unsaturated fat, and it will kind of push all the other phospholipids out of the way. So the point of these different structures is to help the cell survive in different temperatures, because bacteria aren't like us. We strictly control the environment of all of our cells, the temperature, the water, the salinity, all that stuff. Bacteria like E. coli can be in the nice gut of a warm cow 1 second, and then literally the next second they're in a patty on the ground in the cold and the snow. So they have to be able to quickly adapt to these changing environments. So in response to cold shock, that poop coming out of the cow and landing in the snow, they're going to produce more unsaturated fats. If the membrane is super stiff and it's cold, nothing's getting in that membrane. But if the membrane is really fluid in the colder temperatures, molecules can get in and out a lot easier. But we don't want our membrane to be too melty, because then it might kind of fall apart and let too many, things in that we don't want in. So when it gets hotter, we add more of the unsaturated fats and stiffen up the membrane. So in extreme temperature stress, we make these rings, these 3-carbon rings, to make it even more stiff. Sorry, I think I said the other way. This makes it more stiff than the unsaturated fat. It does not increase space. Sorry, I misspoke there. All right, any questions about that? Moving in the same direction for a moment here. You've probably seen molecules like this before in pictures when you were taking cell bio or something like that. Do you remember what we called that molecule in a eukaryotic cell? Cholesterol. I thought I heard it somewhere. So in our cells, we will add these planar molecules, these planar lipids, as a way to help stiffen our membrane. And ours, we add cholesterol. For these guys, they add hopanes or hopanoids. Very similar in structure, but they are a different chemical technically. And the whole job of these is to decrease fluidity. We're stiffening up the membrane here. So we will add more of these in hotter environments. Any questions? Yeah.
The cholesterol and opioids work in
Mm-hmm. But it's hopanoids in bacteria and cholesterol in our cells, eukaryotes. Cool. All right. Archaea, as I said, like to live in really, really extreme environments. So they do some other really cool things to their membrane lipids to help cope with the really, really high heats that they live in. So one thing that they do is that ester bond that originally connects the fatty acid tails to the head of the phospholipid, they replace it with an ether bond. And instead of having that carboxyl shape, it just has an oxygen in between the 2 carbons. This gives the water fewer places to attack and dissolve. So this makes that bond much more stable in an aqueous environment. So just allows for more tolerance and higher temperatures. Then they can make these things called terpenoids. which are these branched chains of hydrocarbons that have methyl groups sticking out of them. And those methyl groups help to limit the movement of the hydrocarbons. It kind of like makes it more stiff and less fluid. So more stiffness means better heat tolerance. Sometimes then we even take the two phospholipids and say, you know what? We're not going to keep these as two molecules anymore. We're going to connect them. And now this is all just going to be one molecule. No longer are there two leaflets, there's just one. And that helps them tolerate extreme temperatures a lot better as well. And then they can even adapt that and create these ring structures in there. And those act similar to the hopanoids. They help stiffen up the membrane even more. So archaea have these really crazy adaptations that just help them live in the super extreme environments that they live in, like the deep sea vents that are super, super high, boiling temperatures and very high pressure. So how can they live at boiling temperatures? Well, they make all these adjustments to their membranes to keep it from dissolving away. Any questions about that? Okay, whew. Good. Now let's move on to the more important stuff of the day. All right, let's talk about the cell envelope. In this case, we're really basically talking about the cell wall, but there's other structures that surround the cell wall, and that's why we call it the envelope. It includes all these structures. It is the most common structural support, the cell wall, and pretty much all bacteria have this. All prokaryotes have a cell wall, except for a few in the family or the group Mycoplasma. These ones have a cell membrane with no outer layers because they are obligate intracellular pathogens, meaning they don't really live in the environment ever. Their home is inside our cells where we keep it nice and comfortable all the time. So they don't need the cell wall and the envelope to keep them protected because we keep them protected as they slowly kill us. But basically, everything else has this cell wall and an envelope in some manner. So let's talk about the cell wall. This is the key thing that we want to take away from this lecture. It's probably the key thing for the first third of your exam. So the cell wall is very flexible, but also very rigid. And it has a lot of holes in it, so we kind of think of it as a mesh bag. And it has to have holes because we still need to get food in, and we still need to get our waste out somehow. If it was solid, nothing could get in and out, and we'd starve to death. Or we'd die from all of our waste products. So the job of it is to provide structural support, but mainly to protect the cell from that osmotic pressure. So as the water comes in and it's pushing out on the membrane, the cell membrane keeps it from lysing. In bacteria, the cell wall is made of peptidoglycan. It's only found in bacteria, so it's a really great target for antibiotics because only bacteria have it, nothing else does. We also call it murin. So peptidoglycan, if we break down that word, peptide refers to what? What four biomolecules? Which one of them? Proteins, yep. And glycan, which one do you think that refers to? No, that's more peptide. The peptide bond connects the amino acid. Think about like gly, carbs, yeah. So these are basically carbs and small proteins together in one molecule. Murin, this just comes from the Latin word with the base muro, like wall. It's the wall chemical. All of these chemicals get linked together. So in the end, all the subunits of peptidoglycan connect to each other, and the whole cell wall becomes one single molecule. That just helps make it even more stable for the cell. If there's any breaks inside the cell wall, that's a target for the cell to be destroyed. That's a weak point in the cell's armor. So they need to connect it all in order to keep it from becoming weak and being a liability. All right, so let's talk about how we make this structure. So first, we'll talk about the glycan chains. These are repeated disaccharides. Di means two, saccharide means sugar. So this is a monosaccharide. Put two together, you now have a disaccharide. So we have these two consisting of these two sugars, N-acetylglucosamine, we also refer to that as NAG or GLICNAC, and N-acetylmuramic acid, NAM or MURNAC. You do unfortunately have to know these terms. So I know these suck, but know those two terms. Now, NAM will be the one that connects to the protein aspect of it, to the small peptide. What we mean by a peptide is it's just a few amino acids long. It's not a full-on protein. Now, these little protein, little peptides that it connects to are what connects one chain of all of these disaccharides to another. So what are we looking at here exactly? We have repeated nag, nam, nag, nam, nag, blah, 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 and they make a long chain. Right next to it, you will have a second long chain, and then a third, and this will wrap all the way around the cell. But we want to connect these. So the NAMs can connect to each other using that little protein. And that brings these two chains together and holds them stable. And all of this is running parallel to the cell membrane. So this is the cell membrane going around, the cell wall comes right on top of it. So let's talk about this peptide portion of it now. We got to review one chemistry thing, and that is chirality. You don't have to understand it deeply. You just have to know that with our amino acids and other chemicals, they have a handedness to them. Like if you look at our hands, there's no way that you can put them together and overlap them for them to be identical to each other. Even if I go like this, I'm seeing the top of one hand and the bottom of another. So they're not identical. And so this creates mirror images, a D form and an L form. Now, all of the proteins for all living things on this planet exclusively use the L form for some reason. We just said we like this one best. Bacteria, though, use a couple D forms to make their peptidoglycan. It's really unique in all of life for these organisms to be using the D form. So it's a really kind of interesting key point here that makes them different from eukaryotes. Ways that you can remember this, actually, the D is from the Latin word for right. So if you think of the Spanish word derecho for right, it's right, L for left. That helps you remember it in the future. Now, you do have to know the peptide, all the amino acids in it in order starting from NAM. So we have our long glycan chain, and from NAM, we start to connect this 5 amino acid peptide. The first one is L-alanine. Then we have a D-glutamic acid. Then the third one is M-diaminopimelic acid. This is technically a precursor of lysine. So it's still an amino acid. It's just an earlier form of it. And then we have 2 D-alanines, so the opposite form of what life normally likes to use. We have 2 D-alanines stuck together. And then the other glycan chain up at the top also has an L-alanine, a D-glutamic acid, an M-diaminopimelic acid, and 2 D-alanines connected to each other. The third, this diaminopimelic acid, is going to connect to the 4th D-alanine of the one across from it. And when that happens, it kicks off the last D-alanine. So we start with 5 amino acids. We make a bond between the 2 different peptides, and when we do, we kick off one of our amino acids. So now we only have 4. So that last D-alanine is cleaved and lost. Does that make sense? Any questions? Yeah. No. Good question. I am not that mean. I'm sure there is some professor out there who is that mean. That's just cruel. We are not chemists here. We are biologists, damn it. All right, so moving on with this. This is just an explanation of what I just said in words. I do that sometimes, I'm sorry. I'll explain it and then I'm like, and then here it is written out for you to take home and study from your slides. All of this is catalyzed by an enzyme group called the transpeptidases. We're making a bond across trans, that's what trans means, the two peptide groups. And we're linking the two peptides together with transpeptidases. So now what this does, I'm going to go back for a second here. We've just taken 4 and 4 amino acids and connected them together to create that cross-link. So really, this NAM sticks out with 4 amino acids, this NAM sticks out with 4 amino acids, and they connect to each other. And that's what is called a cross bridge. Okay. So there's some cool things that happen because of this. The enzymes responsible, as I said, are the transpeptidases. We also call them penicillin binding proteins. That's the other name that they have, because penicillin, the antibiotic, binds to those proteins and keeps them from working. So here's an example. The enzyme, the transpeptidase, the penicillin binding protein, is trying to make that cross bridge. But penicillin comes in and fits in the active site of that enzyme. What type of inhibition is that if it fits in the active site? Competitive inhibition, yes, because it's competing for the substrate in the active site. When the active site is filled, this enzyme cannot come and bind to the peptides. It can't facilitate that cross-link. And so these two peptides don't bond together. And now our peptidoglycan pieces are not connected. And now the cell is weak, and that internal turgor pressure can cause the cell to explode. So that's what penicillin does. It keeps us from forming those connections in the cell wall and makes the cell vulnerable to lysis. So it doesn't directly kill the cell, it makes the cell vulnerable to water, and water kills the cell. Then there's another antibiotic called vancomycin. What that one does, it really, really loves the fact that there's 2 dealanines on the unbound peptide. The dealanine right at the end. It will bind to the 2 of those. And it will prevent this protein, this enzyme, from getting in there and making the cross bridge. Because it's like, I need this. I need to get right here to make a bond between these things, and I need to kick one of them off, and you're still holding it here. So because of that, the bacteria, once again, can't form the cross bridges. They're weak, and the cell explodes due to turgor pressure. So unfortunately, penicillin and vancomycin have a lot of resistance mechanisms against them. We'll talk about more of them in the end. But some bacteria evolved to where instead of a second dealanine, they have a different amino acid there. So vancomycin can't bind it anymore, and they can still make their whole peptidoglycan molecule. They can make those cross bridges. So any questions about the way these 2 antibiotics work? No, they don't kill the cell actively. They don't help us kill the cell any better. All it does is it prevents them from connecting all the pieces of their cell wall together, and it makes them vulnerable to lysis. Yep, good questions. All right. So here we see those penicillin binding proteins, the transpeptidases, and they're working on the peptidoglycan. Now, to make all of the disaccharides come together in the long chain, the glycan chain, we use transglycosylases. Those just link the sugars together. And the synthesis of this is only going to occur where the cell is actively elongating. So only at the points where the cell is growing do we have to come in and fill it in with new peptidoglycan. So it's basically making it where you only need to worry about one weak area at a time, and you can fix it right away. As the cell grows a little bit, you immediately fill in the hole. And the way the cell wall extends is because of a protein called MREB. It's this little guy on the inside that's anchoring this whole complex of how we make the peptidoglycan together. We're going to talk more about MREB in a second, so don't worry too much about it right now. So just know the transglycosylases link the glycan chain together, and that synthesis can only occur where the cell is actively growing. Synthesis of peptidoglycan. All right. Now, because of the way peptidoglycan is made and where the cell prefers to grow, we end up with different shapes of bacterial cells. This is a little bit from chapter 2. So if you're like, I read chapter 3 and this isn't in there, this is just quickly from chapter 2. We have coccus shapes. This is one. Cocci is the plural. These are just spheres. Then we have bacillus or bacilli. We tend to call these rods. Then you have spirochetes. Wait, this one down here. You have similar ones called sporillum. These are not the same technically. And those just form spiral-shaped cells. Then we have the selenomids. These are crescent shapes. And the keyword here, selene, means moon. So if it's a crescent, it's like the moon. And then you have vibrios or comma-shaped. How those differ from the crescent-shaped ones? Good question. Don't really know the answer to that one. It's like not my area of expertise, and I couldn't find a good answer on it, so I'm sorry. But they are technically different. And those are just some examples of species that fall into those that you may have heard of. Staph, like a MRSA infection or a staph infection, streptococcus, like strep throat, E. coli and salmonella, I think we all know those. Borrelia burgdorferi causes Lyme disease from the tick. Caulobacter crescentus is a major crescent-shaped one that we use and study a lot. And Vibrio cholerae is the cholera-causing bacteria. Yuck. So, where do we grow our peptidoglycan? How do the cells prefer to grow? For things like our rods, they're going to grow in zones because, hold on one second, the MREBs, what they do is they actually kind of start in one spot and coil around the cell. So it looks like a spring inside the cell. And wherever they touch the membrane is where we're going to have the membrane or the cell grow and where we have to make our peptidoglycan. Our cocci, they grow from the middle out only. So they're only going to grow in that one zone. And then some species will only grow at an end. So they'll pick a side and they'll just elongate it that way. One of my previous professors described it as like a tube of lipstick and the lipstick is coming out. If that helps you. All righty. I'm going to keep going. Any questions? So most bacteria have additional envelope layers that help provide the structure and the support. And that we break them down into gram-positive, gram-negative, and acid-fast. We're not really going to get into acid-fast quite as much. Gram-positive and gram-negative are the main ones that you have to know, and you got to know them in detail. So let's get into their main differences here. On the left, we have a gram-positive envelope. On the right is the gram-negative envelope. So if we look at the gram-positive, we have the cell membrane, because every cell needs a cell membrane. And then we have a very, very thick layer of peptidoglycan. So it's not just one or two layers. It's not like one or two sheets. It's many stacked on top of each other outside of the membrane. These are connected together by what we call teichoic acids. I'll talk about those more in a minute. Then they will have an S layer, which can have many sugars attached to it as well. Then for gram-negatives, so this is going to be like your staph bacteria. This is going to be like your E. coli, your salmonella. This is the inner membrane, the plasma membrane. We now call it the inner membrane because on the other side of the cell wall is a second membrane. We call it the outer membrane. They only have one, maybe two layers of peptidoglycan. So their cell wall is very, very thin. And the other key thing that they have is this molecule here called LPS, lipopolysaccharide. We'll get more into that in a second. I'm just giving you an overview right now. And this is a very interesting molecule. Now, this space between the inner and the outer membrane where we have our cell wall, we call that the periplasm. It's kind of like the same structure as the cytoplasm, and it has a lot of dissolved materials in it. It's a very, very active site in the cell. And it just has a different name called the periplasm. All right, so let's get into the key things that you have to know about this. So obviously know, main difference, one membrane, really thick cell wall. 2 membranes, very, very thin cell wall. That's the key difference between a gram-positive and a gram-negative. The cell wall size and how many membranes they have. Yeah. 1 membrane, 2 membranes. Yep. Thick cell wall, thin cell wall. This is why I sweat so much. I'm like, I can't stand still when I teach. All right, the S layer. This is found in both. Oh, I'm sorry, yeah. Yes, it does not, because this technically is the cytoplasm, and all of this is just in the external environment. Think of it as just like an outer coating that's in the environment. Yep. This is still the inside of the cell. All of this is the external environment now. So the S layer, found in both of them, is only found in wild cells. Meaning, if I were to take some cells from the environment or from my skin and then put them on a plate in the lab, after we culture them like 2-3 times, that S layer will go away and never come back. So, because of that, it's very hard to study because we can't keep it around in the lab, but it must mean it has some type of protection for it in the environment. It's protecting them from external environmental forces that obviously aren't around in the lab condition. Maybe it's only around when they're in the presence of other species of bacteria. We don't really know for sure. We're still studying it. But it's basically just a really thick crystal layer of protein or sugar proteins. And we think it might help from osmotic stress. Like I said, we're still studying it. All you just have to know is it's found in wild bacteria, and it's found in both of them. All right, ready? Now let's talk about the major important thing in gram-positives, and that's those teichoic acids. So because there's many, many, many layers, all the layers of peptidoglycan will also form cross bridges vertically towards each other, as well as horizontally towards each other, like we learned. But every layer is also connected by these teichoic acids right here. This is their structure. And it's linked by really strong covalent bonds. And they will extend these from the peptidoglycan, the cell wall, all the way down to the lipids of the outer leaflet of the membrane. And what that does is it anchors the cell wall to the membrane itself, so it doesn't like get washed away in the environment. So that's kind of the point of the teichoic acids. Links everything together and anchors the cell wall to the membrane. Any questions? I'm gonna try to go a little bit faster here, I'm sorry. So gram-negative cells have a very unique outer membrane. And this is kind of the fun part of gram-negative cells. They do have that periplasm. Yeah, sorry, I'm making sure I'm on the right one. And we call this the inner membrane. The regular cell membrane is the inner membrane, and this one is the outer membrane. And there's 3 main structures in the outer membrane that you have to know. Lipoproteins, lipopolysaccharide or lipopolysaccharide, LPS, and porins. So we're going to go through these one at a time now. All right, first, lipoproteins. We have a protein in here that we call murine lipoprotein, wall lipoprotein. It's about 65 amino acids long. And what it does is it binds to one of the little peptides on our peptidoglycan, and then also binds to the membrane of the, or the inner leaflet of the outer membrane. What this does is it anchors the cell wall to the outer membrane. So that's the whole point here. This lysine forms a link, basically the same type of cross-link to that 3rd amino acid. And then the cysteine forms the bond to the lipids, and now they're connected together. That outer membrane is connected to the wall, and they're not going anywhere. Next thing is the lipopolysaccharide, which has 3 parts to it. The first one is lipid A. It's this part right here. And the way your book describes it is a 6-legged giraffe, which is strangely right on the nose. We have 6 hydrocarbon chains down here at the bottom, and they're connected to this big complex here that you don't really have to know. They're just basically amino sugars. And this whole structure right down here, the one that is embedded in the membrane, that is lipid A. Because it's hidden in the bacteria, if you just get exposed to the bacteria, you're fine. But if your body says, that's a bacteria, it's a foreign invader, let's attack it, and you destroy that cell, now this part that was safely embedded in the membrane gets released into your body. And our body sees this and freaks out. And it causes gram-negative sepsis, which is a state of shock in our bodies, where all of your blood vessels will dilate simultaneously, and your blood pressure will drop so instantaneously that you can die within seconds to minutes. So this thing is highly reactive with our immune system. So it makes it difficult then, if we're trying to treat you with an infection, we don't want you to be exposed to this because it could kill you. So how do we kill these bacteria without releasing the lipid A is a problem that a lot of doctors have had to solve. We're not going to get into that too much here. That's more in a medical school class. Then we have the core polysaccharide. That's this portion right here on this main picture. It's this part right here on the little picture. And it's just a highly variable repeated unit of sugars. Could be really long, could be kind of shorter. It just depends on the species. And that's depending on, you know, where they live and their environment and all that. Then we have the final piece, the O antigen. This is another long repeated polysaccharide. It can be longer than the cell is long. So these can be incredibly long sugars. And what they do is they can bind to magnesium, and that basically acts as a crosslink. Just like the peptides did for our cell wall, magnesium binds all of these O antigens together and makes this portion of the envelope very rigid and secure. And a cool thing about this too is it helps resist phagocytosis. So white blood cells will see this and they're like, not quite sure how I can get my mouth around that whole thing, so I don't know how to swallow this guy anymore. So basically it keeps them from being attacked from our white blood cells. All right, I'm sorry, I know I keep going. We got a little bit more to cover still. The other part of the outer membrane are those porins that I remembered, or that I mentioned way at the beginning. These are basically just holes in the membrane. They allow nutrients in, waste out, and they aren't super specific. So a lot of our membrane transporters will only bind to one molecule, like just glucose. This one, it's literally a hole. It'll allow pretty much whatever it wants through. Some of them produce more specificity than others, but for the most part, something like this, which is called OMP-F, it's a major one in E. coli, it's just going to allow whatever it wants through. Now, you might say, well, that's kind of dangerous. You just have these holes that let anything through whenever they want. Well, let's say your environment gets super toxic. We just stop producing these. The bacteria will say, okay, we're going to take them out of the membrane, and we're not going to make as many for a while. Then when it gets safe again, they'll start putting more back in. Bacteria are constantly making adjustments based on what's happening in their environment. They can respond nearly immediately to things, and we'll talk about that more later. Okay, acid fast, the third type of envelope. This is crazy. I will admit that. It's very, very crazy. I want you to notice two things about it. First, that dark blue, light blue structure, that's peptidoglycan. So pretty much every single bacterial species and most archaea have peptidoglycan. So it's still there amidst all this other crazy stuff. The majority of this stuff, they are lipids. They are very long lipids. When we get long lipids, they create waxes. So in particular, there's a lipid called a mycolic acid, which is right here. And that is a really strong wax. What that means is not a lot of stuff is getting in and out of this cell. If you wax something, you do it because you don't want water getting in and out. Well, how do we get food in? How do we get waste out? We attach it to water. So if water can't move in and out very easily, these cells grow very, very, very slowly. They're a bitch to study in the lab because my experiments, I could do an experiment in a day. You want to do one experiment with these guys, it can take a month because they grow so slowly. So, but because they grow slowly, they can evade the host defenses. They can be very, very hard to treat because if they're not growing, most antibiotics won't work on them. So these guys evade the immune system really well with all these waxes, and they're very hard to treat. So all you have to know, there's mycolic acids, they're waxes, they make it really hard for things to get in and out. They still have peptidoglycan, and they grow slowly. You will not have to draw this or label this at all. Okay, last parts. I know, I'm sorry, guys. There's a lot to talk about. I even cut a lot from this chapter. I think I'm going to have to start cutting a little bit more, unfortunately. I have to be really picky with what we talk about. All right. The bacterial species also have cytoskeletons. And the types of cytoskeletal proteins they have differ based on the shape. So every single bacteria has FTCZ, FTSZ, FITZ. This forms a protein Z ring around the central point of all cells. So in something like a coccus bacteria, this is the only cytoskeleton they will have. Then MREB gets added, so it's a second protein that rods will have. So they still have FTCZ, FITZ in the middle, FTSZ, excuse me, and they also have MREB. Then the cells that are crescent shaped will add a third cytoskeleton called CRE-S, crescentin. And it just causes that rod to bend a little bit and make the crescent shape. Now, FITZ is the important one because it tells the cell, this is where you're going to start dividing. The rest of them just help perform structural support duties, and this one helps, MREB helps the complex form to make our peptidoglycan. So those are the three cytoskeletal proteins. Okay. Now, quickly talk about cell division. I'm not going to hit this too hard. The most important thing about this is DNA replication, which we are going to talk about when we get to the genetics. So I'm not sure how I'm going to test you on this yet, but it's going to be very minimal, just to let you know. So I'm going to go through it kind of fast. They do not do mitosis or meiosis, bacteria. They undergo what we call binary fission or septation. Meiosis they don't do because they don't reproduce asexually, or they don't reproduce sexually. They just kind of divide. So this is a result of highly coordinated efforts in the cell. Their cell division and their DNA replication have to be bang, bang, bang perfectly in time in order for this to happen properly. So it's going to happen while the cell is actively replicating its DNA. It's really cool. So let's talk about it with some pictures here. So we divide these bacteria by septation. Where that FITZ ring was, that's where we start to form the septum. What it does is it starts to grow in towards the cell until finally it connects, and we've just separated the two cells from each other. And it connects to this huge devisome. It's a big complex of all these different proteins that help you divide the cell. That's about all I need you to know about that. It includes FITZ, it includes our penicillin binding proteins. The rest of them, they're just there. That's all we have to know about them. So how does this happen? The DNA is in the nucleoid. The origin of replication is attached to our phospholipids in the membrane, so it's anchored in. But because there's no nucleus around the DNA, bacteria do something super, super cool. As the cell is actively dividing its DNA, it is also actively doing transcription. RNA polymerases are binding and making the little mRNAs. Ribosomes then immediately attach to the RNAs as they're being formed and start making the protein. So the mRNA can still be growing, and there can be 20 ribosomes attached to it, all in different stages of making the protein. This is how bacteria can respond to their environment so fast. They immediately see there's acid stress, make the proteins to deal with that immediately. So it's really cool, and we'll talk more about that later. But because that's all happening at the same time, division is happening at the same time. So what happens is the chromosome starts to replicate, it duplicates its origin, and the origin starts to separate from itself immediately, the two origins. As this is dividing, the Fitz-Z ring starts to form. As soon as DNA replication finishes, as soon as it hits the termination site, the septum forms and the cell starts dividing. But while that's happening, we've already started a new round of replication. So the cell is constantly just replicate, divide, replicate, divide. That's how bacteria can grow so fast. That's about all you need to know for the exam on this one. We're not going to get any deeper than that at this time. Now, based on where they divide, it creates what we call arrangements. So some bacteria, they say, I'm always putting my septum this way is north, this way is south, and I'm always going to divide that way. So as a result, we end up with our bacteria growing in lines from each other. We call that strepto. Sometimes it'll say, I don't care, I'll divide this way, then I'll divide this way, then this way. That creates clusters that we call staphylo. And then sometimes they'll reproduce in perpendicular planes. So sometimes X, sometimes Y, back to X, back to Y. And that can create tetrads or sarcinae. So we add this to the shape of the cell. We can have streptococci. We can have streptobacilli. We can have staphylococci. We don't really have staphylobacilli. We have diplobacilli, we have palisades, we have things like that. But the idea is these give a hint about how they're arranged. And that's more important for the lab. You'll see that, and we'll review it a lot in the lab. Okay. Whew. I'm going to do one more slide, and then we're going to stop. The rest of it is stuff we're going to talk about later on anyway, so it's okay if we don't get to it right now. So cell polarity just means that often, even though the cell from the outside might look like it's equal everywhere, it might have structures on one end that it doesn't have on the other. The most common of that is called a chemoreceptor. A chemoreceptor is basically like a nose. Our noses and our mouths are chemoreceptors. We're taking in chemicals from our surroundings and smelling and tasting them. So it will often have a chemoreceptor on one side and a tail or flagella on the other. So it can follow its nose and propel itself to wherever the food is. And so different structures happen on different poles for different purposes to help the cell survive its environment better. That's the main point of polarity that I wanted you to get. All right. That is it. We'll mention the rest of those later. There are a bunch of study questions for this one. If you have questions about any of these, just message me and I can help you work through them. We can talk about it in office hours, whatever works best for you guys. Any questions? That was a lot. I'm sorry. Thanks for being patient, guys. All righty.